We were very excited by this Rhes finding, and this study came from a great collaboration with a group from Ken Kosick looking at experimental models. They found in rodents that this Rhes pathway was very involved with enhancing tau pathology. And when they use a drug that actually is already approved for clinical use in cancer, they could stop the process. But what they didn't know, and no one knew because no one looked into this, is how important Rhes was in human brains. And this is what my lab is pretty good in doing, in looking at human brains. So the first thing we did was to check human brains and see if Rhes was present or not, and where it was present. And we did it both with immunostaining, looking directly at the protein in the cells, but also with this single nucleus RNA sequencing that I just commented before. And we could show that Rhes is present in all the neurons. But more than this, because we did the microscopy, we knew that Rhes were present in the cytoplasm of the neurons in a very granular way. And then when looking at disease cases, we started to observe that some of these neurons that should have this Rhes in this granular distribution actually had Rhes in a different distribution, either in very large dots, we call puncta, or it would completely disappear from the cytoplasm and be present only in the dendrites. So we put this study together to understand what kind of neurons are those with these changes. And it turned out that these are the neurons that start to accumulate tau. So it shows a very strong correlation. And to try to check this idea again, we compare these changes in Rhes in the neurons with deposit of synuclein and TDP. There are other neuropathological changes associated with neurodegenerative disease. And then synuclein and TDP doesn't do anything to the cell concerning Rhes. So, it seems to be very tau specific. And on the top of it, we look again at the single nuclear RNA-seq data, and we could demonstrate, and this is not published yet, that the regions or the class of neurons that we found to be most vulnerable to Alzheimer's disease are exactly those that have the biggest change in Rhes expression along AD progression. So what does it mean to us? So first of all, it means that this pathway that involves Rhes seems to be important in the pathogenesis, so create opportunities to even use these drugs that's already out there to try to modify it as a way to maybe modify the disease to be seen. Also show us that the autophagy system, and Rhes is involved with autophagy regulation, seems really to be important in Alzheimer's disease, meaning that efforts to improve or increase the efficiency of autophagy as we age, because when we age, the autophagy system starts to fail, might be useful, again, to prevent Alzheimer's disease plaques and tangles to accumulate. And finally, there are some studies in experimental models from other groups suggesting that Rhes has even another function, which is to create mini tunnels linking neurons. And through these mini tunnels, they can exchange mitochondria, and they might exchange lysosomes, which are important for cell energy and cell for cleaning the trash. So if Rhes is affected and these tunnels are destroyed, so it's another reason to create a neuronal dysfunction. And again, another reason to try to invest more in understanding how we can modulate Rhes in the human brain.